And it's a real pleasure to be here. Thank you for the invitation. But as always, if anybody knows me speaking, I will jump straight in because I have plenty of slides. Now let's look at this picture as a scientist. If you're a biochemist, you look at this, you'll just go, that's a stained structure of the inside of a cell. We're looking at all of these fiber networks. And you can see the intermediate filaments, and they're giving the strength to this cell. In here would be the nucleus. Now, as a chemist, when you look at this, it's something slightly different. Because then it's really, what do we get from this? We have inside the cell the microtubuli, the intermediate filaments, the microfilaments. But break that down, let's deconstruct that. They are all nanometers in size. They are all self-assembled, and they're all helical. That's the first thing we get out of them. They have a diameter 6 to 8 nanometers, the actin filaments, 25 nanometers, 10 nanometers for the intermediate filaments. Now, that's quite interesting, but what really interested me, which suddenly hit me, was, but look how stiff they are. LP is this measure of stiffness, so how straight are they? DNA is a stiffness of, say, 65, 68, double-stranded DNA, 68 nanometers. Yeah, actin filament is 15,000 nanometers stiff. Microtubuli, even stiffer. The intermediate filaments, 1 micrometer. Why would nature make such stiff filaments? Because inherently stiffness leads to insolubility. And these are the filaments on the inside of a cell, and yet on the outside we have exactly the same type of family. We have collagen and fibronectin, fibrinogen. And these are also incredibly stiff, micrometers stiff, self-assembled, helical bundles, incredibly stiff. And they actually are fundamentally important for directing cells, because these stiff collagen fibers, or fibers on the outside, are connected via the integrin to the actin filaments on the inside. So a cell feels what's going on the outside. It feels its surroundings, and that information is mechanically transduced to the inside of the cell. Now, we all know this idea of stem cells, and people say, well, we can direct stem cells, we can send them this direction. There's a big science, can we actually direct them? And this idea that you can take a human mesenchymal stem cell and you can make it into a variety of different types of fat cells, bone cells, neurocells, by simply directing it, by controlling it with, say, a specific growth factor, or even by controlling it by the matrix it's embedded in. The general theory, as of, say, last year, is that if you want to grow a bone cell, you need a stiff material. If you want to grow a neuron, you need a soft material. And somewhere in between for a muscle cell, an intermediate material. I will put my neck out and say 99% of all the science published on that by these famous people is wrong. You don't need that. You actually do not. That is completely wrong. So now I've put my head on the chopping block. I have to try and prove to you why I'm telling the truth. Now, this is the theory, and how it works is that the material on the outside, the cell is, imagine it like a seatbelt. You pull on it, and as you pull on it, it will suddenly fight back. The cell has motors inside which are creating an internal stress, and it's then linked through integrin bindings to the materials on the outside, and as it feels the strain, this triggers, these stress fibers triggers different chemical pathways of YAP and TAZ and RA and RO, which then basically the upregulated and downregulated of different proteins, depending upon the mechanics of the extracellular matrix on the outside, tell the nucleus to go and make something different. So if that's it, why can't we do it? Why is it so difficult to try and direct these stem cells? And the problem lies in, let's go and look at these materials. If I look at a polymer material, a synthetic polymer material, and here we have its stiffness as a function of me applying strain, it's linear. I take this sort of polyacrylamide and 99% of other polymers and I pull them, and basically it has this linear relationship. If I go to fibrin, collagen, actin, and even matrigel, which is used for growing cells, they all have a unique property. They have something called strain stiffening. So when the cells pull on it, the material suddenly becomes incredibly stiff. There are no synthetic materials which show this strain stiffening properly. There are none, yet. And what's more interesting about this is that this strain stiffening is in an incredibly low region, which is called the biological accessible region. Cells can't apply a lot of force. They can only apply a tiny amount of force. A few pascals, less than 10 pascals. Yet all the the natural fibers, collagen, fibrin, actin, neurofilaments, they all basically, this is the stiffness. So they're very, very soft gels. But as soon as the cell pulls on them, they will go up in stiffness by 2 or 3 orders of magnitude in this incredibly very low region. Whereas your normal polymers don't change at all. There are 1 or 2 tri-block polymers which do show strain stiffening, but you have to apply hundreds or thousands of pascals before they start to show it. So this is relatively unique, and nature uses this to control the material. So this would be linear elasticity, no effect, but nature basically has this what's called non-linear elasticity. And it controls, by cross-linking and bundling, the initial stiffness, the amount of force which is required before it suddenly becomes stiff, and what we would call the gradient. How quickly does it become stiffer? And this is what we would like to mimic. Because if you look at Matrigel, which many of you would use in the lab for growing cells, or collagen, both of these show strain stiffening. When you look at them, they're completely different. So this was our hypothesis. How could we make a material which mimics collagen? How would you do that? Where would you start from first principles to build this? You go to a physicist. Because the physicists have pretty much modeled everything. In this case, it was actin, and it's called a semi-flexible polymer network. So if you have a polymer or a bundle of materials where the stiffness is basically 10 times larger than the pore size, when you pull on it, the physics says it will exhibit strain stiffening. And so your stiffness of your material, and this will come back to this sort of nomenclature, is simply dependent upon the stiffness of the bundles divided by the pore size. LC is the pore size. So in principle, if you can make a very stiff material and you can cross-link it such that the pore size is basically 10 times smaller, you would end up with a soft material which should, when you pull it, fight back. So how do you do that from chemistry? The idea was you basically need to make a stiff polymer, but we don't have stiff polymers. We don't make polymers which are 100 nanometers in stiffness. We don't do it. You do double-stranded DNA would be the most natural one, or carbon nanotubes 100 nanometers, but not polymers. But we designed, a long time ago, polyisocyanides, and that's simply an alanine, which when you polymerize it, it forms a helical structure. As it starts to grow, it goes 1, 2, 3, 4, and the fifth one is now on top of the first one, so it starts to form a helical polymer. And this you can make left-handed, and this you can make right-handed. And then we stabilize this by a hydrogen bonding network, a bit like Velcro on the outside of these polymers. And so you can make these polymers, we can grow them almost monodispersed in length, and they are like proteins. You can unfold them by warming them up and refold them. You can add TFA to break up the hydrogen bonds, add triethylamine, and they'll return. They're a thermodynamic helical minimum, like a protein. And they are incredibly stiff. In fact, this simple one, which is a methyl-methyl ester, is 1.5 times stiffer than double-stranded DNA. They have a diameter of roughly 2 nanometers, and they form these single polymers. And not only can we make, this one's relatively small, we can make single polymers which will have a molecular weight of roughly 20 million to 30 million Daltons. A single molecule, which is 20 micrometers long, with a stiffness 1.5 times as stiff as DNA. And we can control this stiffness. We can put functionality on it. We can measure, in this case, this actually has pyrrolines on for electron transfer. We can make single polymers. This is 3 to 4 nanometers thick and several micrometers thick, and then excited here measure energy and electron transfer down a single wire. This molecule itself has a persistence length of 100 nanometers. We can then make one additional hydrogen bond, a tripeptide, and then you can see that, because if any of you go and take polymers and you would normally put them on a surface or in organic solution, they wrap up into a ball. These don't. These polymers have a molecular weight probably around 5 million, and they form extremely stiff. This stiffness is 150 nanometers. So are they really stiff? What would you do with something which is so stiff? Well, we use them for electronics, and I put this in for you, this market, actually. We actually used it for decorating it for electronics because it's a four-fold helical helter-skelter structure where a lot of work went into this, where the chromophores are arranged in a four-fold symmetry. It's extremely crystalline, almost one-dimensional wire, where you can get conductivity down a single wire of 10 to the minus 2, which is exceptional for an organic molecule. And we built them into solar cells. This is a solar cell, roughly 20 nanometers thick. This is the donor, this is our polymer, and you can make them. So that's what we do. We have a perfect scaffold. But what would you do with that? Well, what you can do is the first thing we thought about was, well, how would we measure the stiffness? They don't wrap up into a ball. So if any of you are aware of polymer physics, Diffusion, a long time ago for the Nobel Prize, said, well, if you imagine a polymer network, you imagine a material, it's made up of all these polymers, and effectively they're like worms. They're all entangled within each other. And this one polymer, can only occupy a certain tube of space because it's constricted by its family of brothers and sisters. Yet we can't look at that because there are no synthetic polymers which are stiff enough. So what we did was we built our own confocal microscope with stochastic excitation. We take a drop of our material in there, of our polymers, where we have one which is labeled with dye molecules. And then we measure it. And this is 50 times real speed, the motion of a single molecule, single polymer, moving through a gel of the same polymers. And it's moving 50 times real speed. And what it's doing is it's trying to move along. And actually the only reason it's moving is when I put the top glass plate on, I created a tiny shear which caused it to move. But we can see it because it doesn't wrap up into a ball, it stays as a worm. And as it's moving this way down this page, all these other polymers in front of it are also crossing it. So it reaches a barrier, it has to fold back on itself to find a firefinder route down here. Now, this is really nice that you can measure single molecules in real time, effectively moving through a matrix. And we use this to look at motion inside a cell of a single polymer. However, this is a really appalling experiment because the resolution of this experiment is roughly 200 nanometers. So let's go one step further. What we can do is we say, well, let's actually zoom in now, and let's do the experiment with stochastic excitation and zoom down to 40 nanometers. And this is what we would see. So in this gel is roughly basically 1 milligram per mil of the material. And this is what you would actually see. It's moving in this is a simple Brownian motion of a polymer, not doing a lot, just hanging around there, moving backwards and forwards in and out of focus. If I increase the concentration threefold, this is what it looks like. It's not very exciting, is it? You'd agree. Come on, there's a feedback. It doesn't make you jump out and go, wow. However, let's look at this more closely at a 40 nanometer resolution. Because if we look at this dilute matrix, so what we're going to do is we're going to say we're going to have our dye molecules and we're going to look at the motion of these polymers and say, okay, with high resolution, what's the end-to-end distance? How does it jump both laterally or orthogonally? And measure this motion as a function of time and see where is this polymer we just saw, what space is it exploring? So you saw those two pictures before, then look at it again. That first one you saw, this is the volume of space it occupies. It's Brownian motion, it's moving around all over the place. As soon as we increase the concentration threefold, the same polymer only occupies this space. Because all the other polymers around it in the concentration basically confine it to this tube, this what's called Diffusion tube of space it can occupy because it's constrained by its brothers and sisters. And you can zoom into this. You can see for the very first time for us that really a polymer in a matrix of still very low concentration is constrained to only move in this tube. A polymer is not going all over the place. It's just stuck there. And this is all it's got. It's like being crowded in a train in Japan. That's the only space you're allowed to give by the other people. And you can measure this tube as a function of time. So what you can do is you can say, well, this looks a complicated slide. But what we can do is we can say, well, listen, let's look at this polymer moving, and let's look in a fraction of a second, and then let's look a second later. And if you've ever played that game with children, where one person stands like this and sees if the children are running from the wall, do you know this game? And you suddenly turn around and see how far they've gone. What you can do is you do this at very short time scales, and the polymer has hardly moved. It's basically doing what's a Rouse movement, so it's diffusion. It's just diffusing a little bit along this tube, and it's not even having time to feel all the other molecules nearby. If you wait a little bit longer, what you suddenly see is, wow, this displacement suddenly changes. There is a kink. And at this point, we're now actually seeing it doing reptation. So we can now measure after, say, 0.2 of a second, we're now actually to see this molecule moving through this tube. And this polymer here is basically here, and then after a certain point it jumps out, and then it jumps down, and then it jumps to another place, and it basically is confined in this tube, and then it will jump out and form another tube, and then it will jump out and form another tube. And this is really important for understanding how polymers move through the cytoskeleton inside a cell. They don't just do diffusion, they do reptation, something completely different. So from this measurement, we can see that the entanglement of these polymers is dependent on the polymer length. If it's longer, it has more entanglement. The tube diameter, which is actually 110 nanometers, that's all this, is completely independent of its length. It doesn't matter how long my polymer is, or my other polymers around it, it just feels the stiffness. It feels the stiffness of the polymers around, and those stiffnesses control the tube diameter. So the volume and speed it can move depends on the stiffness of the polymers there. And the connectivity to form this gel is basically that one molecule is feeling 14 others. And so I challenge you, I would love to have some DNA molecules and matrices to measure reptation through to see how that tube diameter would change as a function of the concentration of the DNA. And from that we can actually measure that this polymer is indeed as stiff as we thought it was, because we could also measure the persistence length, which was the goal of this experiment. It's 97 nanometers, 100 nanometers stiff. And so what you really can measure is, because this molecule is so stiff, you can actually measure its motion with a 30 to 40 nanometer resolution and realize that polymers in gels don't move in free diffusion. They move in the Dijen confined tube motion, which is particularly interesting for us for gene therapy, because how do we use these molecules? If anybody actually watched the first lecture, the first introductory lecture, we actually use this for gene therapy. It's now, after 2 years, we discovered it. It's now in clinical trials because it's so effective. And this would also be a use for DNAs. You need a linear polymer which has antibodies on such that it can bind, has multivalency, which gives you incredibly high efficacy, but then has still flexibility that it can actually come back in to allow you clustering. And if anybody does the DNA, you have a fantastic molecule because you can get the same effective molarity, same effective drug delivery for about 1,000 to 1,000,000 less in the drug by doing it this way. So that's actually how we use it to control stem cell differentiation. So the stiffness plays a role. Is it still stiff though? Well, there's one other aspect about this molecule, and we go and do the standard force experiments. We can simply put this molecule on a surface, and we built a confocal AFM so we can pull a single molecule at the same time as looking at the confocal so the fluorescent spectroscopy. And we pull these single molecules, and as many of you know, basically as you pull the cantilever up, the basically force curve and the bend in the force curve allows you to measure the stiffness of the polymer. So we would take our polymers, and we have a distribution of them, and we would pull them all overnight automatically. We would then fit this to what's called an extended worm-like chain molecule, where the force of the tip is related to the stiffness of the molecule and the polymer length. And lo and behold, we can measure the stiffness of these molecules where it's basically 17. That molecule I just told you, which was 97 nanometers, and which we saw on the surface was 76 nanometers, when we pull it is only 17 nanometers. If we put a pi-pi stacking, it's 54 nanometers. If we go to a tripeptide, 156 nanometers. In fact, we can make a range of polymers up to 400 to 500 nanometers stiff, but their solubility drops off as you get more stiff polymers. But why is this molecule so not as stiff as we thought it was? And the problem is because it's a spring. So we take one of these polymers and we pull it, but we don't let go. And then we pull it again, and we don't let go, and we pull it again. And lo and behold, what we see is as we start to pull it, it basically gets less and less and less stiff. And if any of you have played with when you were a student, you took out your pen and you had that spring inside your pen and you pulled it out by mistake. If any of you did that? You have a pen and then it doesn't fit back into the pen. That's effectively what we're doing. We're taking this molecule, and if we pull it, what we see is effectively as we pull it, its stiffness disappears. And that doesn't surprise you, because when you model this, we've got a helix. We've got a 5/1 helix, or 4/1 helix. And what we do is so that the fifth molecule is on top of the first. As we pull it, because it's a helix, it has to twist because of the torsion. So as you pull it, what it does it stretches by 35%, and then it basically makes the hydrogen bond again, but now with a 3/1 helix. If you pull it further, and the energy between these two forms is relatively low, if you pull it further, then lo and behold, you suddenly come to a 2/1 helix, but you tear it apart, and it can't refold it. So what we're doing is we're pulling the spring, we pull too much, and basically that's why we basically reduce the stiffness of the molecule, because in our particular polymer, we have basically this 4/1, this 3/1 in equilibrium, but if we pull it too far, it breaks. We had a problem here. We were trying to use these molecules. I had a contract to make banknotes, which is a great job as an academic because you may get some money from this. And the idea was to use these molecules as security measures in the €50 banknote because of its unique circular dichroism. So we needed to make them water soluble. And so we had a student who was making them water soluble, and we simply said, well, listen, do what any chemist does. Let's put an ethylene glycol tail on and make them water soluble. We make these, and they're still too stiff. They don't dissolve. We make this one, it dissolves very well. We make this one, and it won't dissolve. And we didn't understand why. So we actually did genuinely put it in the fridge and went to the bar and had a drink. We came back the next day, and it's completely soluble. So this is an LCST polymer. It dissolves when it's cold and gelates when it's warm. So it's the reverse. And there are one or two families of molecules like this. So you take it out of the fridge, and it will immediately gelate. And it will gelate to body temperature, whatever you want. But what's unique about this is it gelates 2 orders of magnitude less than any other material in the world. It will gelate 0.0625 milligrams per, so that's 99.99 weight percent water, and it will form a gel. And to give you an analogy, you have a bag of sugar about 1.5 kilos, that will gelate a swimming pool, 250,000 litres. So that's the eureka moment as a scientist to say, well, what's going on? Let's look at it. And when you look at this material, this is what it forms. It forms a network. This hole here is 150 nanometers. So the concentration, by varying the concentration, you will vary the pore size, and the pore size will vary from 50 to 240 nanometers. If you vary the concentration, the bundle size will not change. That is decided by the helicity of the molecule. When this starts to bundle, as you start to get the bigger bundles, the strain involved in twisting it in the helix means that it has a finite structure. You can increase the concentration 500-fold. It will still only form bundles of 6 to 9, because that is the energy minimum. And it's incredibly monodispersed. This is a TEM of it. You take this molecule and you'll see it forms this roughly, these are about 8 nanometers wide, bundles of fibers. But it gelates. So this will be a rheology for people. This is the stiffness of the material. This is temperature. If we have the 3-ethylene glycol and we warm it up, bang, in 1 or 2 degrees it goes to a solid. If we put 4-ethylene glycol there, we warm it up, bang, 42 degrees, it becomes a solid. If you mix the two together, they don't talk to each other, because as soon as this gelates, it ignores that. And if you want to control the temperature of gelation, which we do for in vivo applications, we simply make an average. The average number of ethylene glycols decides the temperature it will gelate. So if you have the 3, it will gelate here. If you have the 4, it will gelate there. If you mix them in a linear relationship, they will gelate here, here, and here. If you mix the 4 and the 9, it will be there. So we can control the temperature within half a degree of which it gelates. Even by salt, it will gelate. If you add cations, potassium, sodium, magnesium, it has no effect. If you add anions, however, you can make the temperature drop down to 10 degrees or the temperature to go up to 40 degrees, because it's simply the Hofmeister series. You simply add salt, it makes the polymer more hydrophobic, and it drops out earlier. So we have these stiff molecules, as stiff as DNA. You basically have them in water. As you warm it up, entropically it squeezes out the water. The polymer becomes more and more hydrophobic. It tries to precipitate out, but it can't, because it forms an entangled network, and you end up with a 3-dimensional gel. But is this anything to do with collagen? So let's go back to some simple mathematics. Now, the simple mathematics says that mice gel, if it's like collagen, should have a relationship between the stiffness, the bundle size, and the pore size. But what is the stiffness of the bundle? I don't know. And how can I work that out? Because theory goes, if I have a helix, the stiffness of a helix is dependent upon the stiffness of a single fibre multiplied by the number of fibres in the bundle, squared. That's known mechanics. So we go off and measure the stiffness of these single polymers in water. And what we do is we measure the stiffness as a function of warming them up, and we can see that these polymers are not very stiff. They're 2 to 20 nanometers stiff. However, as soon as they bundle, what we see is the stiffness increases, because the bundling, the stiffness is 5.6 nanometers, the bundle is 460 nanometers stiff. And that's why you have to have a bundle. A bundle allows you to get very, very stiff fibers from relatively flexible polymers because of the n-squared relationship. And if we plug that in, according to our theory, we should have, because we know what the stiffness is, we know what the stiffness of the polymer, we know the pore size, we know all of these things. In principle, it says, theory says you should have 9 fibers per bundle to give you that gel. And we observe 7. So we really do have a material which mimics nature. Why do I think that's interesting for DNA people? Well, what's interesting here is, if you think about this, if you could increase the bundling size or you can increase the stiffness, in principle, you can make materials as solid as glass, which are 99.99% water. So if you took a DNA which is relatively long, and you could induce it to form bigger bundles, you would suddenly, and had a cross-linking which was relatively small pore sizes, you would suddenly access a DNA material which would be as hard as glass, but would only have micrograms of DNA in, if you follow the theory. I challenge anybody to do that, because the theory said that should be doable. So you can actually say, well, listen, I can't make polymers very, very stiff, but if I make them bundle more, so instead of 16 in a bundle, but 20 in a bundle, then I can access hugely stiff materials. But does it show strain stiffening? If it's a real natural material, do we get strain stiffening? This is what you would see for the natural fibers. This would be strain stiffening. You pull on them, and then at a certain point they become stiff. The answer is yes. We take these materials, and because they have that stiffness and that pore size, when you pull them, they fight back. They suddenly become very, very stiff. Depending on the temperature, this is low temperature, with the stiffness, the gelation is higher at different temperatures, suddenly they become stiff. But this is something really, really interesting from nature. Because if we take these materials and we vary the concentration, you would say, well, listen, you change the concentration, the material becomes less stiff. We agree? Less material, a weaker gel. That's not true with semi-flexible polymer materials, because this gel here is quite stiff, and when I pull it, it becomes even stiffer. This gel down here is almost fluid, because the concentration is 10-fold less. But when I pull it at 10 pascals, the material has exactly the same stiffness as a material which is 10 times more concentrated. So what does that mean is you can make a material just as stiff, but 100 times less concentrated if you apply stress to it. Think about that. If you apply stress to a network, you can make something just as stiff, but with a fraction of the concentration because of the physics of strain stiffening networks. And that's why nature uses it, because if the collagen concentration is lower or whatever, when it applies that force, it will still experience the same stiffness. And that would not happen with a normal material. But does this work for growing cells? Ideally, what we'd like to do is replace Matrigel, which is also this. Can we control cells with this material? Well, where is it compared to nature? This is the picture again. In blue is the natural materials, 10 pascals, and it will suddenly become stiff. This is our material. It's the only synthetic material in the world which mimics the strain stiffening of the natural systems. But does it work? Well, yes, it does. We simply take our cells and we put them in the cool liquid with the cofactors and the food and all the different still gelates in that. We warm it up, and the cells are entrapped in a 3-dimensional network. We then can grow them, and if we want to extract them, we don't have to do a digest or triptidite or break up the network. We simply cool the liquid down, it becomes a liquid again, and we extract the cells. And so in this way, we actually can grow cells extremely well. It's non-toxic. We do it in vivo in mice and people now for regenerative medicine. And it really works. However, there is no interaction between the material and cells. Cells don't feel it. So we have to decorate the cells, and we can easily do that. The chemistry is really trivial. We simply have a polymer which has ethylene glycols, and every now and again we have an azide, and we simply do strain-click chemistry. And we can add proteins, peptides, antibodies, DNAs, we've added, you name it, we've added it without any problem, and we can simply decorate it every 5 nanometers, 10 nanometers, 20 nanometers, 30 nanometers, 40 nanometers, whatever we want to. And it works. This is what you would use to grow fibrin. So this would be in fibrin, you grow these cells, these are smooth muscle cells, and basically this is to grow, if you have a heart attack, this is to grow the vein network on the outside of a heart afterwards. If you have our polymer, it doesn't talk to the cells because the cells basically ignore it. They said, I've got no attachment point, so they just stick together to form a big bundle. And if you start taking a material, then you start to see these veins growing inside this gel, this synthetic gel. Not very impressive. But we can see it more clearly here. We start to end up with this huge growing of veins and networks and cells. We can grow small kidneys. We then simply cool the material down, the network falls away, and we just extract the cells. And that works really well. However, what has this got to do with strain stiffening? So we spent 2 years hard work. Well, listen, I sent people into the lab for 2 years hard work, and they did all the hard work, let's be honest here, and said, okay, we have this strain stiffening, but how can we control it? Can we control how much work a cell would have to do to make it stiff? And we were lucky. Because if we plot the polymer stiffness as a function of length, if the polymers are too stiff, too small, they can't form a network. Once they're long enough, they form a network, and then the stiffness of the network is independent of the polymer length. However, the strain stiffening, the amount of work a cell has to do before it becomes stiffer, gets less and less and less as the polymer gets shorter. So that means we can suddenly access a material where, looks a complicated picture, where we can actually say, listen, we can make a material where the stiffness of the material is the same, but the elasticity is different. And that's the first time you can decouple those two. And you say, listen, everybody says stiffness is important for growing cells. I say, no, it's not. What's important is how much force does the cell have to do to make the material suddenly become stiff. So we said, okay, if we can do this, we can have a material which has a consistent stiffness but has a different elasticity. Consistent stiffness, different elasticity, and see what happens. So, let's take some stem cells. We take our polymer, we decorate it. Every 5 nanometers or 10 nanometers, we're going to put a functional group. We'll then do click chemistry. We will add to this our spacer, add our protein, which will bind to the cells. We then make a series of polymers, where effectively we're increasing the length. The length goes from 182 nanometers, 226 nanometers, to 434 nanometers in length. Every 14 to 15 nanometers, there is a protein. And when you measure the mechanics and the rheology, you would apply 9 pascals, will make the material come stiff, and in the long ones, 9 to 19 pascals. That's all we're talking about. The material is all soft, 300 pascals. Not 30,000, not 100,000, 300 pascals, and this is the strain stiffening. And we're going to give it mesenchymal stem cells. And what we're going to do is give these stem cells, and we're going to put into that solutions which could make them fat cells, bone cells, neurocells. We will let it do what it wants. All that we will do is we'll say the gel is the same, the only thing is different is elasticity. And this is what we see. The stem cells decide. If you have a short polymer of 300 pascals, it will form fat cells. As soon as you start making the material more elastic, so the cell has to work harder to pull it, those fat cells become bone cells. The material stiffness has not changed. It's 1 or 2 orders of magnitude less than anything else people use, because it's not about stiffness. It's about how much work does the cell have to do. And lo and behold, by simply changing the polymer length, and hence the strain stiffening, you change the stem cell. And that's a simplified version, because of course, we then go and look inside the cells and say, well, wait a second, is this really true? let's take this cell apart and start looking at proteins and saying, proteins which are involved in the microtubulin, proteins which are involved in this actin, are these proteins changing inside the cell as a function of the polymer length? And lo and behold, that's exactly what you see. The material gel is exactly the same, stiffness. The only thing is the elasticity. Yet this protein, which controls microtubular formation, is not being formed in the strain stiffening material. This one, which forms bone cells, is. So the whole cell is sensing its environment, pulling on it, saying, hey, wait a second, I have to do a little bit more work here, and then the protein makeup inside the cell changes, and the differentiation changes. So this is where we're at. And there's a lot more I've not talked about, but that would be biology. Because what we're saying is that you can control, if you can understand this mechanics and pore size, you can actually control the whole mechanics of stem cells. You can direct cells in any direction you want by controlling the strain stiffening. Because in turn from the strain stiffening, what we actually do is we control the clustering. We control the amount of effort the cell has to do clustering, and this clustering in turn controls the cell differentiation. So what's interesting there, so if you want to express this completely new material, and there are none out there at all, there are no synthetic strain stiffening materials except we're fortunate the first one. What you need to do is a semi-flexible polymer network where the persistence length is about 10 times more than the pore size. And in principle, you have fantastic materials for this. You have materials where you could program them to bundle. You have materials where you could control the pore size. So in principle, you should make highly responsive DNA materials from this very, very easily. And we discovered this about 2 or 3 years ago, and it's gone to, well, sorry, maybe that's a little bit gross, and don't look at this bit then, but this now has real applications. We now use this in drug therapy for immunology because of the stiffness for T-cell activation. We use it for stem cell differentiation and growing, replacing matrix gel, but the biggest one is actually in wound dressings, because the wound dressings, I'll sort of get rid of the picture, okay? The wound dressings are really important that when your wound is closing, many of you might notice, particularly in burn patients, you end up with a scar. And this scar is because as it's closing, the fibrin elasticity goes wrong, and the fibroblasts which go to myofibroblasts, that mechanism goes wrong. By taking this material as a gel, which we pour into a wound, it gelates in the wound. The elasticity means we actually make wound dressings which stop you get scarring when it heals. And that's an amazing thing from simple science that within 2 years it's now on to patients is really exciting, basically by doing fundamental science. And I don't do any of this. This is the group, but in particular this assistant professor who I work with, who's a fantastic rheologist. And I would love to encourage you, any of you who have DNA materials and you want to look at rheology, come and talk to us, because we'd really be happy to talk to you about it. And at that point, I think just on time, I'll stop. Thank you very much.
Questions? Yeah.
Hi, Nick Stephanopoulos from Northwestern.
Yeah.
Beautiful talk, really, really interesting. I had just one question about your differentiation results.
Yes.
When you go from fat to bone,
Yeah.
you seem to skip the intermediate cell types.
Yeah, but that's partly related to the actual, you know you have to give a growth factors, or which can direct them. In the particular experiments there, this was where we saw the biggest differentiation between them. In that one I showed you there, primarily there are just 2 growth factors in there. We actually modify the gel slightly to do neurons, because we would go to a slightly lower, you need a much lower strain stiffening. And in the materials we have, that's 9. Seems to suit best the fat and the bone in that context. But it's interesting to know, because I didn't talk about 50 slides on that if you start turning off these interactions by adding blebustatins, which would break up the interaction with the microtubule, then you don't see this. If you add then integrin antibodies, which will block the interaction with the GRGDS, then what you see is that you don't get bone anymore. It only stays as fat cells, because that's the natural one it wants to form. So you can really control this. It's quite fascinating. Yeah?
Björn by Karolinska Institute.
Yep.
I was wondering about the neuron differentiation, if you had done that.
We're doing that now, and that I can't tell you.
Yeah. Okay.
I'm sorry. I would love to, but I'm not. Maybe afterwards, but I can't. Yep.
So front.
Yeah.
It's just a question about traction as it pertains to a cell.
Yeah.
Presumably the
number of anchor sites is important?
Yeah, yeah, this is really interesting because, let's be honest, 95% of all cell studies have been done on 2D. And there's still a great debate about the interaction between 2D to 3D. So if we take this particular example we did, if we start reducing the GRGDS concentration by diluting it out, what we actually see is very little effect. And that's interesting. So what we think is that we have more a situation where we really have a micro network which is really covering all in 3 dimensions the material, because our pore size is considerably less than collagen, so there is enough anchoring points to do that. So in that context, we get around it. So we are mimicking much of the behavior, because I think the traction is exactly right. And I think what's happening is you need to have enough points where it can pull to give you that clustering, because that clustering is important for the protein pathways inside. Does that answer your question? Partly.
I think so. I was just wondering if you were able to put cells at the interface of 2 types of polymer networks, whether or not you would be able to get interesting differentiation.
Well, this is exactly where we want to go, because the point is that you can build gradients. And of course, if there's a temperature gradient, there's a stiffness gradient. And so what you can see, the goal, of course, is to start growing the cells now. And then if there's a slight temperature gradient, then you get a different type of cell growing on the outside. And that's, in principle, possible. We can do that with some kidney cells, where we get the kidney growing, and then ethereal cells on the outside start to grow if we change the temperature. But the interesting question, because there are so many questions, at what point does a cell make its decision to be something? And I suppose that's our biggest question at the moment, because we don't know.
Take one last question here.
Yep.
So what's the ratio between the length of the polymers you can get and the size of the side chains?
Well, the size of the side chains in principle is anything as big as you want. We can also grow polymers out there and make bottle broth polymer type networks. Normally what we do when we're doing biology, what we want to do is we want to keep the diameter relatively small, because that I didn't talk about, but the biodistribution is fantastic, because if you put it in the body, it doesn't get pulled out. So normally we would make molecules roughly 4 nanometers in diameter, would be our general preference. When we start putting antibodies on, of course, it gets bigger. But for small molecules, 4 nanometers. Now, the length can be anything, depending on the functional group, can be anything up to 20 micrometers for a single molecule, or down to 50 nanometers. We control that by the monomer to catalyst ratio. So we normally work for the water ones 400 or 500 nanometers long, because that keeps the molecular weight roughly quite small, to 2 million. For the organic ones, we can go even larger. Take your pick, basically.
Okay. Thanks very much, Alan.
My
Please
pleasure.
join me in thanking Alan once again.